Okay, Salvador is from VSNI, and I think everyone here knows VSNI from ASReml, Genset, etc. And well, without further ado, the floor is yours.
Just going to time myself. So when I hear about this symposium, I was thinking, I'm not doing much research in G by E by M, so what can I talk about? And it came to my mind that I can talk about the struggles I have at VSN with support and consulting, what it makes me angry when somebody comes with a question about why my analysis is not running, what are the issues with my data, why the software is failing or is crashing. And that's usually like sort of the support questions I have to deal with, but also in part of the consulting. So we have some data sets and it's usually associated with G by E by M or just G by E, and there are issues on the data and things that make me be concerned about the results and also about the interpretability of what I'm getting. So for that reason, I decided to put a few ideas together, a few elements that I have found and a few probably hints about how you can explore this data and how we can change a little bit the way we see them. It's not very technical statistically, but it probably will bring you a little thing about to worry about or see. So I'm just taking the model, the G by E model, the typical G by E model, where I'm expanding with all the possible interactions. And again, here I'm only taking GEM, but we can have year, or we can have location, or we can have even trait if we expand this to multi-trait models. Obviously, when we expand this, we have these three-way ANOVA table, sort of the way we see it, the way we learn it. And it seems a bit difficult for me to kind of interpret all the elements. So I'm just going to start with the typical G by E. And if I just look at G and E expression and an interaction, I have A plus B, A dot B. And that's the very simple model we normally fit in our statistic classes. And we can think about some of these are fixed or random effects, depending on how we interpret them. So my first question was, well, how do I make sure this model that I'm fitting is giving me the right information? And probably the first thing to think is about what are the objectives of this analysis. And most of you, because this is a G by E by M talk, know about many of these aspects. So we want to do the analysis on this because we want to partition the sources of variability. So looking at the variances or the sum of the squares. And that's going to be very relevant, particularly when we have many more interactions into our model. I want to evaluate the amount of G by E and see if it's a concern and make decisions from there. We obviously want to estimate the main effects. And this is one of the main goals that we do this, where we say, I want the mean across all the levels of the environments or the years or whatever you have. Understand patterns and so on. And then we have obviously the inclusion of selecting genotypes for environments that are not being tested or have a form of prediction from those and so on. So there's plenty of objectives where we have this. So I'm not going too much into detail with this. So the data comes to my hands and then I look at the data. And the first thing is, I call it a posteriori because the experiment has been done and the data might come from just a simple study that is well balanced, well designed, but most of the case, it comes from a mix of things. And typical cases are several years, several breeding programs within a breeding company, or even sometimes a company that bought another company and we decide to put everything together. So obviously there is some issues, and the issues I mentioned here, it's the first one is checking the connectivity, and this goes again back to the A plus B plus A two dots B, and do we have a reasonable connectivity? And it's not only, I'm going to talk more about it, but it's not only at the level of do I have the combination of the treatments, but also at the level of the genotype factors in case you're dealing with males and females on a parental analysis. Do I have enough replication to estimate all the effects? This is a bit of a problem because often we deal with single-site analysis. With augmented designs or unreplicated trials. So do I have replication of a genotype in all the possible environments enough to separate the error from the highest order interaction? and often it's not possible, and that also drives our decision. What is the really main objective of the study? And that also allows me to simplify things, throw some things to be ignored, maybe pick a slightly simpler model that gives me a reasonable result instead of trying to complicate my life completely. And also, obviously, we have the last term where we just look for something that is a bit more simple for the objectives of the analysis. Again, this makes a little bit of a distinction between what it is a proper analysis, an operational analysis to make some reading decision, compared to what it is a more academic analysis where we want to disentangle different information. So let me talk about connectivity, because this is the thing that worries me the most. And this is where the majority of the crashes occur. This is an ideal situation of a G by E, where we have 15 genotypes, and then we have 10 environments. I'm using the word E for the environments. And then we have, obviously, every genotype represented on every single one of these dots. I'm assuming I have enough replication to estimate all the effects. And my objective is to get this marginal mean. So in this case, we have the typical concept of orthogonality, so the independence between the genetic effects and the environmental effects. And also, if I have enough replication, I'll be able to estimate the sum of the squares or the effect associated with the G by E of every genotype in every environment. So that sounds very nice. Obviously, some level of imbalance here, it doesn't destroy my analysis. It's relatively, if we have a few dots missing, it's probably going to be okay. But we have other situations, and this is where I illustrate two more cases. This case here, it's more typical, where not all the genotypes are present in all the environments. And so it is not extremely concerning, but it is concerning at this point, particularly when you focus on the marginal means or your estimation of the means. There are several things that affect me here. One of them is going to be obviously different levels of precision, because I have genotypes represented in more or less environments. The other concern I have is that I'm going to have some bias, especially if I'm not using any of the genetic relationships. I will talk a little bit more about it, but if not using the genetic relationships, I'm going to have a bias because, for example, this genotype is only being averaged across two of the environments, which means it's not sampling all the conditions, it's not going to reflect maybe one of these environments that is extreme. And the other thing that happens is that some of these, because they're missing cells, I have an incomplete representation of the G by E. To calculate the variability associated with the G by E, it's only being estimated with the portion of the individuals that are represented there, and those are the ones that are going to contribute or in the sum of the squares, or they are going to contribute in the variance of G by E that I'm going to be estimating. So there's a lot of issues that happens with this. Now, what happens even worse in that particular case is I have fewer, so all those elements that I mentioned before increase. But there is something extra that happens here, which is the confounding. There is a few cases like this genotype here, which is only represented in one environment, and there's another one here that is also only represented in one environment. Are you going to be able to disentangle the genetic effect from the environmental effect? The answer is no, because there's no information from it. Again, if I use some genetic relationships, I might be able to disentangle in some way, but assuming I don't have that, and that means, where does the genetic effect goes to? Does it go to the main effect of genotype? Or does it go to the effect of genotype or environment that is being estimated from the others? And this is one of the things that happens depending on if you use fixed effects or random effects for your genotype and your G by E. If you use fixed effect, it will go to the first term in your model, which is usually G. If you use random effects, it's going to be partitioned according to the variance components that you've been estimated from the rest of the data. Now, is the rest of the data providing you reasonable estimation of the G by E? And this is, again, bringing a bit of the issues. Often the genotypes that are replicated throughout are the checks. Do the checks represent your population to estimate this G by E? Often it's not the case. So then you see that all these complications start to arise when we look at this specific data. But confounding is quite a big issue in this particular case, okay? Now, this is a more extreme case. Now, this is where genotypes are only present in certain environments, and then probably we can think about these are totally disconnected. So in reality, I should probably be analyzing as blocks or as subsets of the analysis. Again, if we have genetic information, we might be able to connect them, but we have a similar situation. By the way, sometimes we have some checks across that allow us to make a connection, but you have to also be very careful that they are, again, a subset of the population with maybe specific conditions that represent what it goes there. So analysis like this sometimes is the ones that gives you very strange genetic G by E, very strange correlations, some weird heritabilities, or maybe because you have a very different sets, kind of like unexpected variances and unexpected heritabilities. Now, back to the slide. I talked a little bit about replication. I think I'm going to go to the next one. Let me just, obviously, because I work with ASReml, I put the model a little bit more in ASReml. And a couple of things I wanted to mention here is, yes, it gets all my, now I'm going back to my model with all my terms, E, M, G, everything. And you can see again, they're all fixed effects. Do we want them as fixed effects, all of them? Probably not. But one of the things that I always push for is, do we want the residual terms to be heterogeneous? And the answer for me is yes, at least at the level of the environments. Because each of those environments has different heritability, I'm going to expect different precision. Now the question is sometimes even goes, do I want each of these environments to be heterogeneous, or do I want it to expand it for a combination of environment management or environment year or environment? How far do you want to get to this heterogeneity? I'm always afraid to having an homogeneity of variances on this analysis because you weight everything equally. The sites with lower heritability are going to use the average heritability. The sites with high heritability are going to use the average heritability. So you penalize the good ones and favor the bad ones. Now let me put some terms into the random part. Normally, obviously, we put the genotype effect, the G by E, and some of the interactions. Now, the thing I don't like about this model is that we have this very strong assumption of a common variance a common genetic variance across all the environments. And again, as I mentioned before, heterogeneity of variances brings another error, and this again is the ones that produces some of these analysis to kind of crash. Ooh. Okay, so I don't have much time. Again, we can put some of these elements on the random, more elements on the random part with different implications. One good trick to use is to actually combine these terms of environment and management into a single factor, which is T. And this is a combination of the factors and allows me to think of this G by E by M model into something more simplistic. It's collapsing the three-dimensional into two dimensions. Now here you see EM1, EM2 representing the different combinations. And this analysis has problems with connectivity, but also better connectivity at the other level of management and environment. Can we expand that model? Yes, I'm going to use a core GH, where it's going to allow me to estimate a correlation between every pair of EM, and that allows me to have a better design of the structure and be able to combine everything. This trick actually is a very good one to allow you to have full flexibility on all the correlations between environments and so on, even if you have some unbalance between treatment and environment, and we use the core GH. So I'm going to go a little bit quick with that. So this is often an incorrect model where we just correlate the environment and the management. And often the other thing we have talked about it is, Daniel just talked about it, about using the factor analytic in order to do an approximation to this big core GH. Now there is one thing I wanted to mention is when we use the genetic components, so the relationship matrix into a sort of an animal model, we lose some of the problems of the connectivity. For example, in the right case, you might have families that are in common. So in that case, it's less likely to have those conflicts, but we still need to look if there is a strong relationship between the different groups of environments. In this case, you have family 1 present on every single of these environments, and then family 2 present everywhere. So you have a good connectivity, but not the same genotype across all the environments, but they are related through their parents, okay? So there are more extensions. Obviously, I talk about G by E by M and other structures. And one of the things I wanted to mention is because of the complexity of these models, as we combine more things, in ASReml, particularly through Arthur Gilmore, has been expanding a few of the capabilities of ASReml to allow for faster analysis, a bit more stable with this reduced rank plus diagonal approach, and also reducing some of the dimension of the matrices. So there'll be something coming from ASReml to facilitate this in the future. And then one of the things I wanted to show in the last extra minute. So it's always better if you find this data, obviously, to be present before the data gets generated, before the experiments get generated. And the main issues are the same that I just mentioned before. Define clearly the objective, kind of think ahead what is the model you're going to try to fit. Think about your population of inference to decide if things are fixed or random. Again, plan with connectivity and check your connectivity at the level of treatment, environment, management, etc., year, whatever, but also check it out through the genetics if you're going to use that. And then allow for some level of imbalance that doesn't destroy your analysis in the future, because some random imbalance or some destruction is going to happen anyway. Some things are not going to work. And a few resources in case you want to get more information about some of these complex models and the uses of the factor analytic structure and so on.
Okay, thanks a lot. Okay, alas, again, one question. Daniela. Yeah, you get the microphone.
Thanks for your presentation. Could you quickly comment on the models using weights? Because many times we get the means and some kind of error measure.
I think
what is your experience?
My experience is that they work very well. I have been comparing them between without weights and with weights, as obviously it reduces the complexity of the analysis. Two things that happen is, the breeding values are the same, everything seems the same, but the standard errors of the breeding values are reduced a little bit. So you have to be wary about that if you're going to do that sort of inference, because it assumes your weights are known without error, in one sense. And the other problematic thing is that you can't calculate heritability in a straightforward manner, because no longer you have the error variance. So, but if you are happy with those things, I think operationally it makes life a lot easier for complex models.
Sounds strange. You make it more complex in a complex model and life becomes easier?
Yes.
Okay.
So you make it